Nature has always been our greatest source of healing, providing the essential compounds used in medicines, nutritional supplements, and personal care. However, traditional plant extraction methods are slow, inefficient, and vulnerable to climate impacts. This leads to frequent supply chain issues, high costs, impurities, and inventory shortages each year. At Fermelanta, we've developed a new way to produce these compounds without the use of plants. Our technology allows us to create custom bacteria strains that replicate nature's complex metabolic pathways. Acting as mini factories, they convert simple raw materials, like sugar, into valuable target substances. By designing effective pathways and introducing selected genes into these specialized strains, we modify their genetic code of instructions. These instructions enable the engineered bacteria to express specific enzymes that drive a series of chemical reactions. With years of experience in genetic engineering, our technology efficiently introduces over 25 genes within a single bacterial cell. This achieves fully functional expression of all transferred genes, including enzymes sourced from other species. Through further tuning and optimization, we enable the production of complex substances that were once difficult or even impossible to create. Unlike plants, our cell factories grow exponentially, multiplying in minutes and producing valuable ingredients in days instead of years. This scalable process can easily generate up to hundreds of kilograms of active ingredients per batch, significantly reducing costs, land use, water consumption, and CO2 emissions. With this groundbreaking approach, we are building resilient and scalable supply chains to meet the world's growing demands for these essential compounds while preserving the natural world.